stories that we've learned from the genomic era in terms of evolutionary biology and speciation has been our increased awareness of the role of gene flow, sort of before, during, and after the speciation process. And of course, one of the signature stories has been humans and Neanderthals. So when we only looked at mitochondrial DNA, this story seemed very neat, very clean. Humans and Neanderthals diverged, never to come back into contact again. But as we started to look at full genomes, we started to realize that the situation was a little more complicated. And in fact, there had been a history of gene flow between these species. So I started thinking about this sort of gene flow issue back during my PhD at UCLA. I was working on a group of jays in the genus Phylogeography, using mitochondrial DNA to look at cryptic diversity really within what at that time was one species. And what we found when we looked at mitochondrial DNA was this, there was a lot of cryptic diversity there. There were deep divergences in very nice, clean, bifurcating phylogeny. So we had 4 main lineages in the Mexican highlands, and no evidence for gene flow between them at all. One of those lineages was so divergent and old that we elevated it to be its own separate species. So now we have transvolcanic jays and we have Mexican jays. And within those Mexican jays up in the north here, you have several quite divergent lineages. So that was very interesting, very cool. I also had some nuclear data to go along with it. And there in the nuclear data, the story was seeming a little bit more complicated. So what I'm showing you here is a structure graph based on 13 microsatellites. And one of the things we saw was between 2 of these lineages of Mexican jays that were not even sister taxa in the phylogeny, there seemed to be some evidence for hybridization or introgression happening between them along some north-south gradient. Another thing we saw was kind of interesting. When we looked at there was one locus in particular of those microsatellites that was kind of weird. In the transvolcanic jays, the allele sizes were very big, highly variable, and anywhere from 280 to like 440 base pairs long. However, in Mexican jays, almost always you found very short alleles. They were like about 100 base pairs long. Except in one place in Mexican jays, and that was right down here by the contact, kind of the suture zone, or not the contact zone, but where the transvolcanic jays and Mexican jays came close to one another. You found these big alleles in Mexican jays, and then decreasing in frequency as you went north. So it was almost like there was some sort of signature here of some past gene flow that had happened. And in fact, by the time you got all the way up into the Chiricahua Mountains in Arizona up there, we sampled maybe 50 individuals. And in fact, there was one individual that was a heterozygote for, it had a small allele and a big allele. So these alleles seem to have been able to spread all the way to the north. You know, that story was too cute not to publish, so we did. And although I think the story's cool, I also think it's kind of funny that there's a paper published based on one microsatellite locus. But you couldn't even see, so the signature was not in the mitochondrial DNA, and you couldn't see it in the phenotype either. So what I'm showing you here is a graph of tail length versus wing length. Transvolcanic jays and Mexican jays are very different in their size. The transvolcanic jays are much bigger. And the dots that are dark are ones that had alleles of the other type. So there actually were a couple of transvolcanic jays that had small alleles, 2 individuals, and a lot of Mexican jays that had the big alleles, but they didn't really look any different from their brethren. So you couldn't see this in the phenotype either. So, all right, time passes. We develop some genomic methods. I get a job a grant. I have a postdoc. And like, you know, with this embarrassment of riches, like many a PI before me, what do? But I go straight back to my PhD study organism. And, you know, but this time with a lot more genomic firepower. This is work that's mainly been done by my postdoc, Eugenia Sarsa. She's been doing a great job. This is part of a large collaborative grant that involves many more species of birds, reptiles, and amphibians. But I'll just be talking about Mexican jays here. So we took a pretty small set of individuals, but we tried to capture the geographic variation. We looked at 14 localities, 20 individuals, and we sampled for over 4,000 UCEs, gathering about 2 million base pairs of data. And I'll be talking about some of these. These are different hypotheses for gene flow, but we'll get into detail with those in a little bit. So jumping right into the results here, one of the nice things you find with UCEs often is that you can collect all of these off-target mitochondrial reads and assemble essentially full mitochondrial genomes just off of off-target bycatch. And so we started with that, and we were happy to find that the phylogeny that we got there was, you know, perfectly matching what we had found before with smaller mitochondrial pieces. And everything looked nice and clean and bifurcating with no gene flow. What I'm showing you here is the concatenated UCE tree. And there's lots of things we could talk about here. You've got the transvolcanic jays that are sister to the Mexican jays in a pretty deep split, just like in the mitochondrial phylogeny. But I want to draw your attention here to one of the main differences between the mitochondrial tree and the nuclear tree is drawing these two lineages together, yellow here and green. I'm sorry, I changed my color scheme between the two papers, which is a bad idea. I still have time to go back and change that. But taking these two lineages here and bringing them together as sister lineages in the UCE tree when they were not in the mitochondrial tree. So this seemed to validate, this discord between mitochondrial and nuclear seemed to validate this prior evidence for gene flow from the microsatellite data. Okay, when we looked at, the next thing we did was we actually drew the informative SNPs out of the UCE loci and just kind of looked at all the SNPs separately, analyzing them in one of these species tree programs that uses SNPs instead of gene trees. So we had a data set of 2,500 SNPs, and we saw a similar pattern where those two lineages were again coming together as sister taxa, but I want to draw your attention here to a different pattern that we hadn't seen before, which was individuals from a particular locality over here on the far eastern edge of this blue western group. These individuals seem to share a significant amount of ancestry with the adjacent green group here that we hadn't seen in any other analysis. So again, this seemed to be some sort of evidence of a hidden history of gene flow that was coming out of these genomic data that we weren't otherwise able to observe. Finally, we wanted to do some more fine-scale hypothesis testing between groups. And so here we turn to something called the ABBA test. This is the portion of the talk where I get to sound like a complete idiot. Say ABBA over again. So I think it's worth explaining a little bit how you do these tests. You're testing for gene flow between non-sister lineages, and you need 4 taxa to do it. One of these is an outgroup. And then you have 3 taxa in your in-group. And this relies on the fact that somewhere in here between the outgroup and the in-group, a mutation has happened, creating a polymorphism. And then if there's been no gene flow and no selection, this polymorphism should sort of sort itself out between the species randomly. And so if your polymorphism is between an A allele and a B allele, you should get equal numbers of these ABBA and VABA patterns in the end, all else being equal, unless there's been gene flow. between these non-sister lineages, in which you would expect an excess of bees flowing from this lineage into this, creating an excess of these ABBA patterns. So that's essentially what this test does. It goes SNP by SNP, assessing the patterns and looking for an elevated number of ABBA patterns relative to BABA patterns. So in terms of the Jays here, we use Western scrub jay as our outgroup. And in this case, if we were testing between transvolcanics and the Western group, we'd be looking for gene flow between these non-sister lineages. So again, ABBA patterns are the ones reflective of gene flow. When we tested what was sort of a positive control here, we had a pretty good idea that there was gene flow happening. We did find highly elevated ABBA patterns. What I'm showing you here, two different numbers. These are the ABBAs on the left. Two different iterations using different individuals. So it wasn't some sort of idiosyncratic thing about the individual. So we felt good about that. We also did a null test, where we just took random individuals from in here and did the test, and we found no elevation, elevated ABBA patterns, so that felt good too. Then looking between this group, where the SNAP tree kind of showed us some evidence, we also found elevated ABBA patterns. So the ABBA test seemed to suggest that there was some gene flow happening there. And then testing this case, where really we only had that one microsatellite locus that really gave that signature of gene flow. ABBA patterns were elevated, but not excessively. We did significance tests on these, and those were close to, but not quite significant. So kind of walking back to the beginning here, the big picture, I mean, think about this. We sampled nearly 400 individuals, and we never found a case of elevated, of mitochondrial DNA that was in the wrong lineage. Not across this divide, not across this divide. And so this gene flow does not seem to be contemporary. It seems to be ancient. Do I think that this is just something about jays or something about hominids? No. I think we're going to find, as we start to look at more systems in a genomic level, that speciation with bouts of gene flow has been more the norm than the exception. So we're just kind of putting all these data together now from the different species in a comparative framework. And very interested if people out there are expert in methods of comparative phylogeography with huge data sets, be interested in talking to you about how exactly we might go about that. So thanks, and I'll take any questions. Yeah.
You don't think it could just be male-mediated gene flow?
So I don't, because the females in Phylogeography jays, all field studies indicate that females are the more highly dispersing sex. They're in very saturated habitats where there's just groups of them, and the females are the ones who kind of have to strike out